Hello, my name is Dr. Roderick L. Roll, and today we will be talking about physical methods of controlling microorganisms. We have already talked about extreme heat. Now we will talk about extreme cold. Refrigeration and freezing will decrease microbial metabolism, growth, and reproduction. It halts growth of most pathogens. Organisms that can live in the presence of this frigid temperature are referred to as sacrophiles. So sacrophiles are cold-loving microorganisms. Several organisms exist that are not affected by extreme cold temperature. Two examples are Listeria and Yersinia. Listeria can be found in refrigerated foods that are held at 5 degrees Celsius, whereas Yersinia can be found in refrigerated blood that is held at 5 degrees Celsius. One very notable example of an outbreak was with bluebell ice cream. Listeria was the organism that was in their ice cream because it is a cold tolerant microorganism. Quick freezing does not kill the pathogen. Slow freezing is more effective at killing the pathogen. It has the ability to form these ice crystals that will puncture the cell membrane as the freezing process is occurring. Extreme temperature from -70 to -135 is inadequate to kill microorganisms. Many bacteria, viruses, and fungus can survive, so therefore it is not good at killing, but it is good at being used as a long-term storage. Many pathogens can survive months of extreme cold. Listed below are several examples. The next method of controlling these microbes that we will discuss is drying out. It's referred to as desiccation. So desiccation will inhibit growth due to the removal of water. This is a microbiostatic environment. The bacteria will not grow, but it's not dead. The following microbes are resistant to desiccation. The following microbes are not resistant to desiccation. Lyophilization is a hybrid between drying out and freezing. So lyophilization is used for long-term storage. Drying out and then freezing is going to prevent these damaging ice crystals that form that will puncture the cell membrane. So this is not an effective method of sterilization. Filtration is another form of inhibiting microbial growth. So we will use different filter sizes. So filtration using 0.22 micrometer pore size will exclude all bacteria. Filtration using 0.01 micrometer pore size will exclude all viruses. This slide here shows you different filters that can be used to exclude different organisms based on size. Filtration is used when the cell media is sensitive to heat. So in cell culturing, we use stuff like fetal bovine serum or horse serum or calf serum. Well, in those solutions or proteins that are required for growth of the cell, if you destroy the structure of the protein, then the cells will not grow. So filtration is used to sterilize this media, to pull all the microbes out of the media, but it will not pull the protein out of the media. How is this achieved is you put the media into this funnel. As it trickles through, percolates through, the media containing the proteins, the growth factors, the antibiotics, if you put it in there, they're going to be in the sterile media, but the microbes that can harm the cell will be restricted or excluded from going through the filter paper. This filter paper can be achieved not only through liquid media, but also through air. So air can be filtered by utilizing filters. One classical example is the HEPA filter. This is used in the hospitals to create these clean rooms. This HEPA filter is 99% efficient at removing these airborne particles. Nurseries, intensive care, and operating rooms utilize these filters. Viruses typically attach to large particles. Therefore, viruses can be captured also. A large particle like dust or water will be trapped in the filter, and attached to that water molecule or that dust molecule is the virus. Now you will utilize this time to color physical control, number 43. The next type of microbial control, physical method, is called osmotic pressure. This is when we utilize salt or sugar to create a hypertonic environment. This is going to restrict bacteria from growing. Remember, in a hypertonic solution, the water is going to be pulled away from the cell. The cell is going to then shrivel. So this will kill or inhibit the cell from growing. We talked about this earlier. In a hypertonic solution, water is going to diffuse out of the cell into the environment to try to decrease the concentration of salt. The removal of the water from the microbe is going to kill the microbe. I would like you to tell me which one is saline, which one is 1% salt, and which one is 10% salt. This is the answer to that question. So with osmotic pressure, we can utilize salt and sugar at a high concentration that is going to create a hypertonic solution. Water is going to be lost from the cell. Therefore, the microbe is going to dry out, desiccation. Fungus have a greater ability than bacteria to survive the hypertonic environment. The next control mechanism for controlling microbes physically is radiation. The shorter the wavelength, the more energy, the greater the penetration power. Radiation can be described in two forms: ionizing radiation and non-ionizing radiation. Ionizing radiation has sufficient energy to eject electrons from atoms, which create ions. Remember, the atom is composed of electrons and protons and neutrons. If electrons leave the atom will become a cation. If electron is gained, then the atom will become an anion. Those are ions. The classical example is salt, which is sodium, which is a cation, and chlorine, which is a anion. Ionizing radiation includes any wavelength that falls below 1 nanometer. So everything to the left of this black line in this image would be ionizing radiation, starting with the most intense energy being the electron beam, followed by the gamma ray, followed by the X-ray. So those are three subtypes that have the ability to physically destroy microbes. The electron beam has the shortest wavelength and are effective at killing, but they do not penetrate well. This can be used to sterilize spices, sterilize meat, microbial plasticware, medical devices, and dental supplies. The next form of ionizing radiation we will talk about is gamma rays. Gamma rays penetrate well, but require hours to kill the microbe. Gamma rays will be used to sterilize meats, spices, and fresh fruits and vegetables. You might have seen this in a grocery store in the produce section. This radura is a symbol that means that that spice or that vegetable or that fruit have been sterilized with gamma rays. The third type is X-rays. So X-rays are used for medical purposes. Now we will talk about non-ionizing radiation. This is going to be to the right of the black line. It will have a wavelength that will be greater than 1 nanometer in diameter. This non-ionizing radiation will affect three-dimensional structure of proteins and nucleic acids. The first one we will talk about is ultraviolet light. UV light does not penetrate well. UV light can cause pyrimidine dimers to be created in DNA. This type of non-ionizing radiation is suitable for disinfecting air, transparent fluids, and surfaces of objects. This is an image of a laminar flow hood. Inside the one to the right, we have the UV light on. It is now disinfecting the air and all of the plastic parts that are inside of this laminar flow hood. Ultraviolet light can also be used to sterilize water, as seen in this water treatment facility plant in the picture. Ultraviolet light can cause cancer because it creates these pyrimidine dimers. So we all know to wear sunscreen and sunblock. This is going to prevent ultraviolet light from penetrating the skin and damaging the DNA. If the DNA is damaged, when DNA replication occurs to create a new cell, the new cell is going to have damaged DNA also. This is referred to as cancer. We talked about the central dogma theory and how a protein is created from the DNA. But if the DNA is damaged, the protein that is created will be misfolded or misshaped. So this summarizes all of the physical methods of controlling microorganisms.